discussion. On respect to Professor George Chalakov and the organizers, and gratitude. I start my lecture. I'm a scientist. I'd rather I'm more into biochemistry, biophysics. I gather most of you are into medicine, right? So I'll try to get to your language later, but to understand what life is per se, this is a paradigm shift talk. Why does life need oxygen and lipids? Murburn concept, my brainchild, gives real answers. So in this, we are all living beings. You can live without food for a few days, for a few days with water also, but oxygen, within a few minutes you die. And I say that you're in a medical university, the world all over. I challenge that nobody knows why they need oxygen, why we need oxygen. And here in this talk, you will hear why we need oxygen. I claim, this is a very radical claim. I am going to challenge much of what you have learned, what you teach, to probabilistic redox fields. My name is Manoj, and I am very honored to represent my institution, Amrita Vishwavidyapitham, and my country, India, and the curious humanity also, the open-minded mindset that is required to indulge new ideas. While inaugurating, she said that we need to have new directions. You bet I am trying to pitch something very radical in this talk. So, let me put the classical clinical realities and perceptions as of 10 years back. The problem is, why is oxygen deemed essential for life? Oxygen is supposed to be the terminal electron acceptor functioning in the mitochondrial electron transport chain. All right? So, but then it also gives rise to reactive oxygen species and oxidative stress. It's supposed to stay bound at complex 4, taking 4 electrons, 4 protons to give 2 molecules of water. So that's rather a very, very passive way of working. And it's a 2-electron reaction. And lipids are rather inert molecules. They are also in chemical energy storage, forming frameworks for embedding proteins. There you see fatty tissue cells. Also, they are also involved in pathological manifestations, obesity, and atherosclerosis. In my slides, the bottom ones, the highlighted ones in yellow, they are the crucial questions or messages. Why does biology paradoxically depend on dangerous molecules, which can give reactive oxygen species and which can clog our systems? Basically, if Ross reactive oxygen species were merely toxic, evolution should have eliminated them. Why are they yet implicated in metabolism, immunity, signaling, and all that? Why is antioxidant therapy largely ineffective in cardiovascular diseases? What if reactive oxygen species and peroxidations are systemic needs and ploys? This is some thought process that we will indulge. So we see that both lipids and oxygen are like Dr. Jekyll and Mr. Hyde. If you, from the earlier times, you will know R.L. Stevenson's twin-faced, identities. And if you're a young generation person, the Two-Face from Batman comics, right? It has got both qualities. Now, some philosophy first, before I get into my talk. A critic is a necessary evil, and criticism is evil necessity, said Carolyn Wells, the American writer. You see, science essentially advances with falsification. It does not work with validation. It works with falsification. So I am a critical thinker, and I would like to falsify more than validate. But then the natural people like to validate, but I'll get to that too. So imagine how science works is, now you have a hypothesis saying that an ant goes in a circular path. Now you take points and you try to plot them, and then suppose if it takes a small violation from that circular path, you have an equation 2 pi r, that's the equation of a circle, you try to plot. If it abides by that, good. But then the problem is, do we have a pointer? Pointer? Where is it? This one. Oopsie. Which one is the light? Okay. So, but if you take points here, 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 and here, always here within the circle, you can get it. Even if the reality is something completely different, it can go out of the circle. You can take points all here and still get a circle if you want to get a circle. So that's something you have to very clearly watch out. And then these points, you can, experimental points, this can be fitted into a Michaelis-Menten type hyperbola, a parabola, an exponential, two-phase, one-phase. Mathematical interpretations are highly, also subjective. So here is Escher's, my favorite picture. You see, the water goes up the second floor, comes, then turns the wheel. Again, at every single point, you see the narrative that we are given. It would look like nice at one point, it appeals, but you put it all together, it doesn't make sense. So this is something also that you need to watch out. So I would argue that many a times what happens is in science, if a theory is bound by limitations of a cube, what we do is we put a cone in and try to incorporate whatever doesn't get into it through this. This is the way in which many times science is taken. So, but here I would critically argue that Karl Popper's philosophy of scientific advancement fundamentally rests on the practice of empirical falsification. So I'm a critical thinker. Please bear with me. So, with all due respect to whatever we have learned, what we have studied, I'll still go on. Now, the classical perspective is what you're taught in the schools, in the medical colleges. This is what you're taught. Basically, cells are the fundamental building blocks of life, and cells have the basic two biological principles. Defining cells is through cell theory, and central dogma is what affords controls. Central dogma gives proteins. Proteins have active sites, and then active sites work based on topology, and then selectivity, specificity, all that results because of these genes coding proteins which have, whether they're enzymes, electron donors, or ligand receptors, they have this topological identification. Like I see Paul, he has got a beard. I can understand how he looks. So same thing at the active site at a molecular level. This is the way they function. So the metabolism is understood as sequential activities. One more like assembly, car assembly. Different parts are assembled. Something like that. Now the predictable kinetics, integral stoichiometry, all that is a feature of the classical biochemical paradigm. So also in this, the sodium-potassium balance and the energetics are dealt with highly effective cation-proton gradients, and they are harnessed for energetics and intelligence. And then all these are multiphasic orchestrations. So several proteins, but then this is the classical paradigm. But then we also know that several proteins are non-specific and membranes are also leaky, and serial sequential reactions with multi-phase spatial coordination. Is it attainable? That's sometimes a difficult thing. So now we know that cells, we are mechanical systems, we are electrical systems, we are chemical systems, we have all these systems involved. The only workable force that we can use out of the four fundamental forces is electromagnetic force. So moving electrons and moieties are the fundamental ways in which we can produce energy or do useful work. So now here you see PChems is powering coherence. How does one cell or a bunch of cells come together to function as a whole, as a unit. And homeostasis, how does it maintain its internal composition vis-a-vis external pressures? And electromechanics, sensing response, how does it take in the cues and how does it respond? Now you see classical textbook, multiple Nobel recognized ideas is this, wherein you have protons in the mitochondria, they are pumped out, they create a gradient, and they come back simultaneously to make ATP. This is electron transport, chain coupled proton pumping, proton motive force is generated, it's called chemiosmosis, and then proton recycle coupled to ATP synthesis. ATP is the energy currency. You take it like you have a currency here, euro, or you take it to any shop, they give you something. Similarly, ATP is supposed to be the energy currency. So in terms of homeostasis, how the cell's membranes, the lipid membranes, measure, estimate, and then translocate sodium, potassium ions using ATP. So classical perceptions deem cell membrane sentient sentience. Sentient is somehow they have some kind of an intelligence, and they can safeguard, they can measure, and they can transport. So ATP as an energy currency, central dogmas, determinism is adequate. So here is where the critical thought process comes in. We will challenge this whole thing. Please bear with me. This criticism is a must. So first thing is something that even a school-going kid could have done before the recognition. That is, mitochondria, you can check it out yourself. Take a notebook, check it out. Mitochondria is a micro-dimension organ. It has got this much water, and that has got this kind of dissociation. So the amount of H plus, the H plus concentration is this much, this is the water, multiplied by Avogadro's number, you should get the exact number of protons in a mitochondria. So that is practically finger countable. Cyanobacterium have practically no protons. So this is not volume adjusted for the internal components, I'm just taking the volume. So basically, you have prokaryotes and mitochondria that are practically aprotic. And then we have the theory of proton motive force, which says that protons are pumped. When there are no protons to pump, how can it pump? If you say water breaks up to give protons, no, it cannot, because that reaction is 79 kilojoules per mole, highly endergonic. So submicron dimensional bioenergetic molecules lack free protons to pump. And protons are 0.00002 angstroms, whereas the smallest atom known is 2 angstroms. So how can it control such a small dimension? Numbers are not there, control mechanisms are not available like that. Then buffering microdomains and proton hopping Grotthuss mechanisms cannot overcome these thermodynamic and spatial constraints. That's the argument. And then the second argument, fundamental electrochemical argument. You maintain a membrane at a potential difference, and then you cannot have ions going in and out in all directions continuously. It's very difficult. The classical electrochemical paradigm tells us that ions will move in a particular way only. Here, if you see electron transport chain, the concept of electron transport chain. Now see, you have a laptop here. How does this laptop work? This is solid. You have printed circuitry, you have potential difference. But our system, it's a fluid system, it's a spongy system, it's a soft system. We don't have critical electrical networks. So how does it work? The classical theory states that we have, either in case of photosynthesis or in case of respiration, we have multiple protein complexes, and electrons go for making one molecule of water or one molecule of NADH, hundreds of electron transfers for one molecule. How could this ever be possible? And then, sorry, how do I go back? Yeah. So in photosynthesis, water is broken down to get NADPH and ATP plus oxygen, whereas in respiration, NAD, the nicotinamide reduced molecule, is used to make water and ATP. So both are present in sometimes in the same system. How can these electronic circuits work when oxygen plus electron, this fundamental reaction is minus 250 kilojoules per mole. Oxygen will mess with every single so-called deterministic circuitry. There is no way. Oxygen is a small, highly mobile diatomic molecule. So serial connectivity that we assume in all these theories are improbable. Proteins are randomly distributed in ratios and space. They are not present in the way the classical theories see. So the polarity will not allow, the electrical polarity will not allow ions migrations in left, right, and center ways. The very important thing, if you have read Stephen Hawking's Brief History of Time, you know he talks about a hot cup of tea dropping from the hand, breaking into pieces. This is a spontaneous reaction. You cannot have the reverse reaction. That is, the pieces of cup, the heat that has dissipated, coming together to give a hot cup of tea. Similarly, if you take a fruit, you can put the fruit through a mixer and rotate it. And now you get juice and pump at the bottom. Now you invert the juicer, or you try to rotate it counterclockwise. You try to put the juice and pulp and try to get the fruit. Is it possible? No. That's what the classical theory says. You have the complex 5 going this way, it uses ATP, and that way it gives ATP. So this is a highly exergonic reaction. Hydrolysis is an exergonic reaction. The formation is an endergonic reaction. So is reversible phosphorylation achievable under physiological conditions to account for the outcomes? Complex 5, the Boyer's model, so which we teach. So I would say, very simply, cyanobacteria have both respiratory and photosynthetic electron transport chains on them. Same membrane. How does cytochrome C, which is a part of both respiratory and photosynthetic electron transport chain, know whether should I make water or should I break water? So the only reaction that is thermodynamically easily feasible is respiration, and photosynthesis would not occur if it were to be the classical theory. And then protons are of course not available in cyanobacteria, and they are too small. Once again, erythrocyte, you see the red blood cells, they have no nucleus, they have no mitochondria, yet they function for 3 to 4 months quite happily. They maintain sodium potassium differential. So how is that possible? There's no genetic control, there's no glycolysis enzymes cannot make the amount of ATP that is needed. So most importantly, plants lack sodium potassium ATPase, the protein that Jensku found, which is supposed to be the protein responsible for creating the sodium potassium gradient. So acclaimed the claims are not tenable or are inadequate, I would say. So here I would like to introduce you to the new theory called Murburn concept. A brainchild. It's based fundamentally on that reactions occur in crowded stochastic environment within the cell, soft water dispersed medium, and diffusible reactive species cannot be avoided in this system, and system level control must be distributed, not localized. So here is a very different set of ideas on which we build. So now I will show you what the theory is, then we'll come to the applications in different systems. Now here the theory, the classical theory is you have an enzyme, you have a substrate, they are topologically complementary, whether electron donor, acceptor, all that, whether for electron transport chain or for catalysis. So in Murburn theorization, there's only one small difference, one small but very, very important difference. That is, the electron transfer or the catalysis can be mediated by a diffusible reactive species or diffusible reactive oxygen species. So murzymes are those kinds of proteins that catalyze these kinds of reactions that involve intermediary diffusible reactive species. So Murburn burning, Murburn burning. Murd is archaic English, which means closed burning. Okay, so a burning reaction, let me tell you, you take a paper, burn it. Take exactly the same paper, set it afire exactly in the same way. You cannot get it to burn in the same way again. So it's a stochastic reaction and it will give different ratios, different profiles. So that's the difference. It's not like a classical enzyme catalyzed reaction where you have very clear cut, same beautiful kinetics, same beautiful stoichiometry. Here it won't be happening. So molecules are found in cells, unbound ions are found in cells, and radicals and radiations are generated or formed in cells. So this is an interactive paradigm which can explain a new types of metabolic physiological phenomena. So molecule unbound radical, mild unrestricted redox catalysis. This is based on effective charge separation. An electron that was resident at one point moves because of a diffusible reactive species formation. So that is the mobilization of electrons. So this is called ECS, and the simple chemical engines are the outcomes of this. That is, once an oxygen can come, oxygen molecule can come and take an electron that was here or on the substrate and move it somewhere else, and thereby effective charge separation can result, and the cell functions as a simple chemical engine. Simple chemical engine. We are simple chemical systems, so that's how it functions. So how do we see mechanistic interpretations in this? That is, now in the classical system, enzyme-protein interactions, you have allosteric modulation or classical competitive site, active site inhibitions, where you have to have binding-based outcomes. Here you will get high selectivities, specific redox partnering, and integral stoichiometry. This is deterministic. But in the Murburn mechanism, because the radicals or reactive species can be formed and they can venture outside in vicinity of the protein, you can get interaction equilibrium between molecules, ions, and radicals in the Mur zone, and then you can have activation and inhibition and alternate product formation, various physiological outcomes, giving rise to broad substrate specificities, non-specific redox partners, and non-integral stoichiometry. Here it is stochastic. So here you see the paradigm again. You have molecules, ions, and substrates, enzyme molecules. You have oxygen, and it forms water in the reductive pathway of oxygen, and diffusible reactive species are formed. There is one electron equilibrium. So it's like a whirlpool from which products are formed. Here, once again, the enzyme substrate or donor acceptor or receptor modulant, their interactions need not be very clearly complementary, but there can be some non-specific, non-selective topological interactions, but yet they can give rise to products. So here, effective charge separation, diffusible reactive species, chemico-electromagnetic matrix. This is yet another principle of Murburn, that is the same formation. It results rather than intelligent pumping and all that. Here it's a chemical electromagnetic matrix which gives to the powering, coherence, homeostasis, electromechanical and sensing response activities. So this is what gives rise to simple chemical engine. Reaction control rather than topological, it's based on delta G of formation, thermodynamic pull, dynamic partitioning, sustained release, concentration-based stabilization, proton spin locks, classical chemical controls. So here, what does Murburn concept at the outset explain? At the outset, it explains how one single enzyme can convert diverse topology molecules. Then, how can diverse molecules give different reactions by a simple enzyme? How can you get multifacial, zeroth order, and biphasic, all these reactions? How can we have Km less than the Kd value, which is theoretically not possible by Michaelis-Menten kinetics? This is something that you will be interested in as physicians. Here, at nanomolar level, there could be good pharmacological activity, but at millimolar level, you might not observe. So this is hormesis. Why? Because if you have a diffusible radical, that can get stabilized at low concentrations. And if it is at higher concentration, they can collapse. So they will give 2 electron outcomes. So this can explain some of the hormetic and idiosyncratic dose responses. Idiosyncratic is how each one person can give different kind of response, and hormetic is when the dose is low, you get a higher pharmacological activity. And then you can also explain how lab-to-lab variations are explained, how uncoupling or loss of redox equivalents are observed, and non-integral reaction ratios. And then, very importantly, how you observe electricity, voltage. Now, the classical theory required you to pump positive ions like sodium, 3 sodium out, and 2 potassium in. Here, oxygen comes in, receives an electron, and becomes negative. So when you take voltage, cells will be negative potential at the resting state. So this is a completely new explanation for a lot of simple physiological or in situ outcomes at the outset. So now that I have shown you what Murburn theory is, what classical theory is, now I'll get into the different applications. Starting with the story of I started working with Lowell Hager, who was a professor in Urbana-Champaign, and this chloroperoxidase was a heme enzyme that he had isolated and worked on for 30 years. So when you give 2 molecules like thiourea and monochlorodiamidone, what happens is the enzyme uses thiourea first. So they said, oh, this specificity or selectivity is inherent in the active site. Now what I showed is, you take the common toilet cleaning bleach molecule, you add that bleach very slowly, 1 molecule by 1 molecule, you get practically 100% selectivity for thiourea. So sustained addition of a radical or a reactive species can give specificity. But if you add large amount, they don't. So suppose if I come from the room and if this is not too crowded, I see Professor Chaldakov and I like him more than others, I will naturally come to him. Something like that, you know. But if there is a lot of other people, I will collide and there's a chance that I'll react with the others. So that's the kind of thing that happens in cells. That is, the reactive species are released in a sustained manner. Though they are very highly reactive, they can still give specificity and selectivity. And I showed that in this enzyme, I differentiated active site processes and diffusible reactive species mediated enzyme reactions. This is an indene molecule. This double bond is attacked by chlorination as well as by epoxidation mechanism. The epoxidation mechanism is dependent upon the substrate concentration, you can see. Whereas the chlorination mechanism is practically zero order. Same molecule, same enzyme, 2 different reactions. One occur outside the active site, mediated by diffusible species, chlorination, whereas the epoxidation reaction, oxygen insertion, can occur at the active site. And now I showed that this reaction, you just manipulate the pH and the salt content, the specificity and selectivity varies. So this specificity and selectivity is not inherent in the enzyme active site. That's a very important thing that experimentally was demonstrated here. My professor, some of you young people, you might experience things on yourself, in your life. When I found this, I reported to my prof and he said, nah, it must be an artifact. And then I was working on it one more day in the night. Azide is a heme enzyme inhibitor, like cyanide. And what happened is, when I reduced,
Go.
Heme proteins are having very similar reactions. Heme proteins are involved in microsomes, the drug metabolizing systems in liver, mitochondria, and the chloroperoxidase, just the system that I showed in Lowell Hager's lab. There you see an active site excluded substrate. If you add, it just depletes the electron source of peroxide. Similarly, you will see the same mechanism in microsomes also. The zeroth order chemistry, how when you add the xenobiotic, that is the drugs that you use, or ADP in mitochondria, it just changes. In all these systems, compound 1 was supposed to be the enzyme-mediated species. Now, here we come to the drug metabolizing system, wherein the cytochrome P450, hundreds of types, very high, the reddish brown color of the liver is because of this. Like red is because of, in the blood, is because of hemoglobin, it's because of cytochrome P450. It's supposed to bind to a reductase, and they are supposed to go through elaborate electron transfer and sequential systems to give the hydroxylation of the drug. Multiple types of reactants. Now here, one simple experiment I did that challenged. Here the theory states that the heme enzyme must bind to the drug molecule, and then to that reductase binds, and then we have protein-protein mediated electron transfer. Oxygen is activated at the cytochrome P450 heme enzyme center to give diffusible reactive species. My project in the NIH was to minimize the reactive oxygen species production in drug optimization. When you try to get a new drug, it goes through a 10 to 15 year cycle. You have to optimize the drug to minimize the reactive oxygen species production. Here, what we showed is very clearly, without the substrate, the peroxide was higher. So the theory, the classical theory that, substrate is required for reactive oxygen species production was clearly refuted. Now, here is how different molecules that are metabolized by CYP2C9, the common NSAIDs, non-steroidal anti-inflammatory drugs, sildenafil, so many, diverse topology, one CYP2C9. How can it all be accommodated? Here you see very clearly, these molecules can bind at different places, and what this enzyme actually does is stabilize the diffusible reactive oxygen species, and different kinds of diffusible reactive oxygen species can interact with the drug metabolism, drug molecule. So CYP3A4, one of the most prolific enzymes, this is trabacted in too large a molecule, it binds outside, and then the reactive oxygen species can easily access it. So when you have substrate, amino acids that are on the surface mutated, you get the complete molecule metabolism changing. The classical theory that it must go into the active site no longer is verified. Now, here we have multiple CYPs. All of their heme thiolate ligands you can see here. They are easily accessible. So they can easily get reduced, and then different molecules can bind to their topology. So this theory, new theory, that reactive oxygen species are activated by reductase, flavin, to give superoxide, which is used by cytochrome P450, explains how you have hundreds of different cytochrome P450, but a unique reductase, flavin reductase, which is at a very low concentration. So the classical theory requires the reductase to bind to all these 450s. It's not possible. So now we come to the most interesting part of this talk, that is the respiration by mitochondria. The classical theory dictates you have the serial electron chain, wherein coenzyme Q is supposed to take 2 electrons. You must have studied in your biochemistry text. Protons are pumped out, and then the complex 5 goes round and round to make using the proton gradient. It's something like hydroelectric power project. So the redox reaction and the phosphorylation reaction are completely different, disconnected. Now, we showed that ADP and phosphate, if you just take it in test tube and add a reactive oxygen species, you get ATP. This reaction is inhibited by cyanide. So simply, it's the reactive oxygen species that is doing the reaction. That is, all these complex 1 through 4, what they do is they activate oxygen to give a diffusible reactive oxygen species, and then they have ADP binding sites, increasing the affinity of the interaction probability of ADP and activated phosphate to interact with each other. So this new theory explains the diverse morphology of mitochondria, how they look. In the classical theory, if the cristae was like this, the complex 1, complex 2, complex 3, they had to be arranged in a particular way. So the new theory is supported by the fact that The more you add NADH and oxygen in mitochondria, you get more diffusible reactive oxygen species. More diffusible oxygen species, more ATP. More diffusible reactive oxygen species, more greater the transmembrane potential. ATP production is not seen without reactive oxygen species, and complex 1 to 4 generate ROS and bind ADP. The minimal requirements of the Murburn theory is completely taken. All right, and so here you have the Murburn mechanism, which is highly exergonic. So this is the reason. Now you see, I will just analyze one protein structure. This is complex 1, which is using NADH. This is a prokaryotic system. This is the eukaryotic system. Now here you see multiple iron sulfur center, flavin center, and this coenzyme Q. Coenzyme Q is somehow supposed to go into the protein like a snake and take the electron there. Now how can it be when this cannot move at any feasible reaction rate? Also, if their theory was true, then we should have had these redox centers in the membrane system, not hanging like this. This is perfectly agreeable with the Murburn model, where ADP binding sites are here. These redox centers are not in absolute beautiful sequence. And most importantly, it explains why NADH is the electron donor. It gives 2 electrons, but it can give only 1 proton. So when that proton is limited, the radical becomes stable, and then you have protons coming from outside. So here you see the reactions, the classical theory, it's endergonic, whereas the Murburn reaction, each complex, it is justifiable. So here is adipose tissue, heat production, brown adipose tissue, how it produces, that is, this uncoupling protein or thermogenins, they have high amount of lysine and arginine inside, what Sensei was talking about yesterday in terms of RAGE. Now, when they are present in the intermembrane state, they can move the reactive oxygen species negatively charged here, and they can easily get protonated. O gets added to H, that's highly thermogenic, and then water forms, and then you have heat formation. So these are very simple thermodynamic explanations supported by kinetic observation. This is the image in Adipobiology 2019, where the paradigm of brown adipose tissue or the fatty acids being obligatorily required for heat generation was changed. Here, the fatty acid was absolutely essential for heat generation. I said that ROS generation is the primary reaction, and then you have multiple outcomes thereafter. I'll go quickly now. So here is the overall paradigm, that is, here the mitochondrion is like a naturally formed hearth, wherein you have the ATP production based on the flames, which is equivalent to DROS. There are analogous. So basically, transmembrane species potential is because of negatively charged species produced inside, not because of proton pumping. I'll quickly go over the remaining slides. This is the most important thing. Every physiology practically in our system is affected by cyanide. The classical theory explains that cyanide must go and bind to the ion center of cytochrome oxidase. So this is a stoichiometric event, 1:1. So the cells have approximately 100 to 1,000 times higher cyanide toxic concentration. So if you have 1-to-1 correlation, then also it cannot explain the dosage. 99 to 99.9% of cytochrome oxidase is free. How can you explain? Then carbon monoxide in smokers, carbon monoxide is present at 40%, and at 10%, and it knocks you out at 40%. So, but cyanide kills at 2.5%. Cyanide is almost, you see the binding constants, kinetic and equilibrium constants for cyanide and carbon monoxide. Carbon monoxide, any chemist would know that it is at least 1,000,000 times a better ligand than cyanide. But yet, we cannot get. And also here also, the Km value is much lower than Kd value. Theoretically, it is not possible. So the Murburn theory explains that cyanide converts the reactive oxygen species as a catalyst, not as a ligand, by converting the superoxide and hydroxyl radical into water. without getting consumed. So this reaction is kinetically and thermodynamically highly facile. So this is the reason why cyanide is toxic in all physiology. So other applications, photosynthesis. I will not go into this because not vision. Andreas wanted to know this. You see the eye, all of you when you study, remember these kind of things. Question. This is the eye anatomy, right? Rods and cones are supposed to be here. The light photon should clearly go through approximately 300 to 400 micron thick retina. The photon wavelength is 400 nanometers. It has to traverse 1,000 times the wavelength. By the time it reaches, and these are all dynamic layers of different structures and optical qualities. So by the time it reaches, one photon from some place will reach some other place. It cannot evolve. So the Murburn model says that oxygen, and you see both sides are highly innervated. The action potential must arise. What happened? This is gone, right? Oh. The action potential must arise here, go like this, and come back to the optical nerve like that. If that were true, the simplest evolutionary structure is this. So I say that oxygen is directly involved, and it helps understand the function of transducin coupled with rhodopsin. The reactive oxygen species openings are very clearly, beautifully structurally available. The beta transducin, which has no role, was predicted to be a GTPase, which is what it is. So I have now solved the, now you see intelligence. In the classical system of, you see potassium sodium pump, neurology, neurobiology. Here it is electronics, here it is ionics. We show that it is actually electronics, and the anion differential is what is giving the transmembrane potential. If you take sodium and potassium, it's approximately 150 millimolar inside, 150 millimolar outside. If we were to go with an Nernst potential, you cannot. So I have also now, this is yet unpublished, single master equation can give all the features of neuronal function. So now here is lactate dehydrogenase. When you exert, the lactate dehydrogenase is supposed to, it is present 4 times higher in your muscle. But then the lactate is supposed to go to liver. Why? Liver has lesser lactate dehydrogenase. They say the classical textbooks teach us that it is reversible. No, this reaction, the textbooks have from the electrochemical and equilibrium point calculation. I have calculated the free energy change starting from the free energy of formation. This reaction is very high. It goes to the liver because there is a high amount of reactive oxygen species by cytochrome P50 and flavin. So it goes through the recycling part in a totally different way. I have justified it based on simple docking, and here electrophysiology. The sodium potassium difference, this is also very important. They say that the smaller ion sodium, if it were inside, we can understand seeding. Mackinaon's model says that somehow potassium, which is the larger ion, is selected. No, you see the thermodynamic signatures. The thermodynamic signature of potassium is very different from that of sodium and potassium. So sodium and proton. So here earlier there were these kind of theories wherein ion absorption and ion pumping. Murburn says that it is effective charge separation. My collaborator showed that even reactive oxygen species can give effective charge separation and membrane potential. And I'm quickly coming to the end. Very beautifully, while the chloride ion is very different, carbonate, bicarbonate, acetate, phosphate, all these, the potassium is more soluble inside the cell. So that is, respiratory anions can help co-solubilize potassium better. There is no need for any pumping. So what cells do is thermodynamically viable. We are not violating anything there. There is no need for any pumping. So hemoglobin, it's a tetramer. There is 2 beta proteins there. They have a pore like this. So iron 3 can dissociate to give superoxide. Superoxide can bind and make ATP. So here you see, so it can give either oxygen or superoxide. So when it gives out superoxide, it can make ATP. So that's what is explaining the tetrameric nature of hemoglobin. The beautiful beta-more has this feature. It was not explained earlier. Cyclooxygen is the, different fatty acids cannot go into the active site. The flagellar mechanical mobility, it's supposed to go rotate. It's very simple. It's just a water ejecting system. And this flagellum, which goes, apparently looks like going like that, it's actually a stabilizer. Like you have a kite, when you fly a kite, the long tail, it's just a stabilizer. It's not going round and round. So it was a misunderstanding. So overviews and wrapping up. Now it is effective charge separation, and it is not transmembrane proton pumps or ion pumps. So Murburn predictions show that DRS levels should correlate with function, not damage alone, which is shown. Lipid phases should modulate redox buffering. So peroxidations and oxidations of lipids is natural. Non-integral stoichiometry should usually be noted. So here we have Murburn at 3 steps: molecular interactions, catalysis, and electron transfer. When it comes to a cellular level, what you suppose, imagine a hydrocarbon goes and oxygen goes, it creates heat and some amount of charge, electrical activity, the pressure of water formation, the water spontaneously goes out and taking up the waste. So this is the way at macroscopic level. At conceptual level, the central dogma deterministic works at a long term, and stochastic short term is by Murburn concept. So here is a theoretical understanding for you. That is, classical theory deems that cells and its contents work with like a orchestra. Every piece is given a definite meter, length, time to play. Whereas the jazz band is, they come together and somehow work it out. So that's the analogy. So you see all these organelles, whether they are rods and cones in eye, cyanobacterial lipid packing in the liver microsomes, or chloroplast, mitochondria, wherever there's a high amount of lipid invagination and packing, it is to minimize water activity so that oxygen-centered radicals are stable in the lipid medium to do their work. All right, so here we explain water is the main solvent and also the reactant. Ions are also for electron conduction. Lipids provide proton limiting interface. Proteins are not just working at active site, they can also work at delocalized site. These are very, very different idea. Carbohydrates have hydroxyl groups because they are products of Murburn through evolution, and nucleic acids are protected on both sides by ribose sugars, hydroxyl groups, and phosphate because Murburn was the primary reason. So oxygen, most importantly, is for generating DRS. It's not for oxygen per se. It is for the radical species, which we now take it to be major oxidative stress and cancer and aging alone. It is a necessary evil. So now here you have spatial reaction site probability, state, transition state probability. Enzymes were the original enzymes in which classical enzymes are a subclass. So the classical theory is not negated by Murzyme or Murburn theory. The Murzyme theory says that it is the earlier one and the classical enzymes came later. So the reactive oxygen species are primarily something that lead to chaos, but they are also something that instill order. If you go to a kitchen, you see a knife. You see knife cutting boards and knife racks also. Just because there are antioxidant enzymes like superoxide dismutase, catalase, glutathione peroxidase, all that, it doesn't mean that reactive oxygen species are not used. Like knife. Knife is absolutely essential. Without knife, you cannot have. So you take a wick, you put it in oil, you burn the wick. What burns? The lipid burns, the oil burns first. That's what happens in our life. We have to die because our system takes the collateral damage of the essential roles of reactive oxygen species. That's why the oxygen is needed. So When we come to school level, we see that combustion and respiration are different, but then here it is very similar. So oxygen is more soluble in lipids, 5 to 6 times more, and therefore lipids are now a non-functionalized proton-limiting DRS rate. Otherwise, carbohydrates could have also been the membrane, or proteins could have, but then lipids are more important because they offer a relatively lesser reactive interface. So now Murburn is completely redefining respiration, photosynthesis, thermogenesis, the fundamental bioenergetic paradigm. If I have to move, I have to talk, I have to walk, I have to make anything, it requires energy. And that fundamental process is through Murburn process. And then how it senses external signals, how electrophysiology, mechanophysiology, ecology, how drug metabolism, lactate, dose responses, all these from toxicity of cyanide, immunology, origin, sustenance, termination, evolution of life, all that, this new theory is absolutely relevant. So what is in it for you? Yeah, fine, yeah, yes. Finally, the mainstreaming of classical, parallel systems, Chinese, homeopathic. I'm not saying it's all right, but please, medical professionals, don't call them fake science and all that. It's not good, because there are so many hit and trial things that they have worked out. A Chinese, acupuncture person can press here, and some discomfort at an alternate site could be relieved. There is a reason for that. Some food in Kerala, we say it's hot. Very clearly, some bananas are hot, some bananas are not. That's because there are some small amount of components in that they can give rise to heat. So traditional and parallel systems of medicines can be mainstream, and then we will have in the future redox metrics and sensors in clinical practice, reinterpretation of clinical experimental data will also happen, personalized therapy in conjunction with artificial intelligence, and novel dietary approaches and lifestyle suggestions, and new aids and prosthetics, because Murburn will allow you to interface. Now electrochemical, electrophysiological aspects are all figured. Now credits. This is my Professor Lowell Haggard. I started this work in 1999 in his lab. Working in his lab, availing his money, I questioned his works. Like Sensei said, first you agree, and then you disagree, and then you move on. He was so nice, he allowed me to stay back, but the others were not so generous. In the land of opportunities, I was asked to move out. So, yeah, they said, you cannot question the main hypothesis in the field, drug metabolism. So this is Stanislav Yanev, who I got introduced in 2016 while my paper was submitted to Frontiers in Pharmacology. Wonderful man. Because of him, I got introduced to senior George. And thanks to George, wonderful person, how much ever I say. And these people are from the science organization in my country. And now I have, like what he did, he constituted a fund to serve the memory of his grandson. I have spoken about him, this was in 2024 in my class, about his decalogue. And you see, I have also constituted a fund, HYCS, Haggard, Yanev, Charlakov, and Soman, the guy, my dean currently, who gave me a job. I worked 10 years without a salary, sitting in my house developing this idea. So these are my collaborators. Nikolai Barin, a thermodynamic guru from Russia. Hiro Tamagawa from Japan, and Lauren Jakin from Belgium, and Yan Yu Wu from China, Daniel Gideon, Abhinav Parashar, Vivian Dekat, David Jacob, Afsal Manikatodi, and Suljit Ramaswamy, my PhD students. Finally, I have this philosophy of looking at research with a very open mind. Rather than being dismissive, entertain a theory first. This is Bill Watterson's childlike mentality shown through Calvin, 6-year-old boy who talks with his stuffed tiger, hopes. So we did not evolve to avoid fire. We evolved to live with controlled fire. That's the fundamental message. You see Charaka Samhita, the medical text from ancient Yor India, it says, Shante Agno Mriyate. If the fire dies out, you die. The fundamental digestion process in Kerala, my place, it's called Dahanam. Dahanam means burning. I don't know how they could think of such a word so long. I came to know about this only recently, a couple of years back. So thank you. So outreach and all those slides.